the company was founded by a neuroscientist and a medicinal chemist who came together towards a common goal. And the goal was to identify a therapeutic that would be beneficial to patients targeting amyloid-beta oligomers. And so what they established was a phenotypic assay, where the assay was set up in a cortical neuronal culture system. It was a phenotypic assay understanding whether or not compounds could restore a trafficking deficit incurred by amyloid-beta oligomers. And from that came a series of molecules that were hits, and that led to the identification through lead optimization to the clinical candidate CT1812. So with regard to the data set we obtained for our Alzheimer's program, most of that has been published, and you'll see that on our website. But the studies range from in vitro to in vivo efficacy, and of course we have our clinical trial data now that has, for some of them that have read out. Starting off in the in vitro system, the neuronal culture system, not only do we know that CT1812 can restore neuronal trafficking deficits incurred by amyloid-beta oligomers, we also have data supporting that CT1812 can block the ability of amyloid-beta oligomers from binding to neuronal synapses, and also can protect neuronal synapses from damage incurred by the amyloid-beta oligomers. And that mostly was elaborated in the neuronal culture system. We also have our in vivo efficacy model. Now, this is an Alzheimer's transgenic animal model where amyloid-beta is present already. And these animals were dosed with a single dose of CT1812 or a vehicle. And what we identified there was that amyloid-beta oligomers were being displaced and elevated in the CSF, suggesting that once the A-beta oligomer is displaced from the neuronal synapses, it makes it out into the CSF. Now, chronic dosing studies in the amyloid or the Alzheimer's transgenic mice, we found that CT1812 dosed over a series of weeks led to restoration of cognitive and memory deficits incurred and that occur in this transgenic Alzheimer's mouse model. And so that was very exciting for us. And then fast forwarding to our clinical trial SNAP, which we published or presented a presentation in Barcelona at the AD/PD meeting in March this year. What we found there was that just like we elaborated in our preclinical Alzheimer's transgenic mouse model, we found that in a small number of patients, those given CT1812 also demonstrated a displacement of the A-beta oligomers into the CSF after a single dose of CT1812, which is supportive of our mechanism of action holding true in the clinic, which is why we're very excited to measure in a longer course trial clinical efficacy and benefit to the patients.
So if you're familiar with Decode Genetics, 1995, Kari Stephenson studied a population in Iceland and was looking, because it was a largely homogeneous population, he was able to look at the genetics of the population. And in a population of people who did not get Alzheimer's, like 4-to-1, what they found was a particular mutation on the amyloid precursor protein. 673, I think, was the position. It's in the title of the paper, but alanine and threonine swap positions. What does that do for you? It means people around you are 4 times more likely to get Alzheimer's disease. So our founders that Mary talked about proceeded to do in vitro work. And what we were able to demonstrate with CT1812, we were able to replicate through pharmacologically targeting, going after the sigma-2 target, the ability to recreate that protective effect in a dish. The displacement of oligomers mimicked that. And as a team, Tony, our CMO, Mary, and I have talked about this many times. How often are you able to drug a target to mimic a protective mutation in nature?